That we can go ahead and get started. I'll just introduce myself really quickly and then we'll get started on the seminar. So my name is Corey Broeckling. I am the director of ARC Biocenter, which is primarily a mass spectrometry center here within ARC, primarily focused on proteomics and metabolomics, but I also do a fair amount of other mass spectrometry type applications. And what I want to talk today about is the use of tandem mass spectrometry. And this is actually almost a bit of a sub-research field. And I thought it was worth its own seminar. And so we can go ahead and get started. I share my slides. So tandem mass spectrometry is really a field of research within the larger field of mass spectrometry. So to understand tandem mass spectrometry, we need to first have a little bit of an understanding of mass spectrometry. I'm not going to go through all of this in detail, but we are going to talk a little bit about mass spectrometry instrumentation before we get to the tandem component. So mass spectrometry is the separation and measurement of ionized molecules by their mass, particularly by their mass to charge ratio, which you will often see abbreviated M/Z. The ultimate output of any mass spectrometry experiment is really a mass spectrum. And on this mass spectrum cartoon that you can see here, we have two axes. One is the mass to charge ratio and the other is the intensity. That mass to charge ratio is, of course, our mass oftentimes, or we can infer mass from mass to charge. And then the intensity is going to be proportional to the molecular concentration that we have introduced into the mass spectrometer. So in an ideal world, then the signal intensity increases with concentration. So we get two properties. One is the mass and one is an intensity. Mass spectrometry isn't a instrument. And I think that for those of you that are new to the field, this is one of the more complicated parts of understanding the world of mass spectrometry is understanding what the different mass analyzers actually are and how they differ. I don't want this to be a talk on mass analyzers, but I need to introduce them because tandem mass spectrometry is really a hybrid type platform, and we're going to need to understand the mass analyzers at some level to understand tandem mass spectrometry. And so we'll talk a little bit about these four mass analyzers: a quadrupole, an ion trap, a time of flight, and an orbitrap. And I use these schematics, these sort of cartoon versions of the instrument, the mass analyzer itself, in many of the figures that we'll see throughout the talk today. So let's first start talking about the quadrupole. The quadrupole is a mass analyzer which operates as an ion filter. And if you look at the large, let me just turn on my pointer here, the large cartoon down here at the bottom, if we have a population of ions that go into a quadrupole, normally what we're trying to do with the quadrupole is to isolate one mass from all of the others. And so what happens is many ions of many masses go into the quadrupole, but only ions with a certain mass come out of the quadrupole. The other ions are discarded, and these ions are never actually detected. So you could have 100 different ion masses go in, but if only one of them is allowed to pass through, all of those other 99 are discarded into the waste, and only the one target one is the one that hits our detector, which is this cartoon back here on the right. And so a quadrupole operates as a filter. It's not exactly measuring mass. It's just that it only allows ions of a certain mass to pass. All the others are sent to waste. It's also important to note that the quadrupole can, in some cases, be used to transmit all ions. So if we have a quadrupole, we can actually use it either to send all the ions through or just the ones we want. And we'll actually see a little bit of that as we get further into the talk. The time of flight is an ion separator. So you'll often see time of flight abbreviated as TOF. So a TOF mass spectrometer is time of flight. And basically, the basic principle here is that you send that same population of ions in front of what's called a pusher or a pulsar, some sort of plate. You apply a voltage, and that accelerates the ions. Now, it accelerates them in a mass-to-charge ratio-dependent velocity. So the lighter the ion, the faster they travel. And this is a separator because we don't discard any of the ions. We let all of them hit the detector. They're just separated in time. And so a time-of-flight instrument is wonderful when you want to see all of the ions in a population, not just the ones that you're primarily interested in. An Orbitrap is another very popular high resolving power, high mass accuracy instrument, and it operates a little bit differently. So it's actually what I describe as an oscillator, and the way that this basically works is you introduce that population of ions into the Orbitrap, and they oscillate around the central electrode. And this is a three-dimensional trap. So imagine sort of the inside of if you hollowed out a donut. That's sort of what it would look like on the inside. And as they oscillate around this central electrode, that oscillation is patterned in a mass-dependent manner. The detector actually sits outside of the Orbitrap over here, and it measures what we call the ion current. As this oscillation occurs, you get ion current that is measured by the indirect detector that looks something like this. You see this pattern up here in this cartoon spectrum above with the red and the green ions. They oscillate, and over this transient, that oscillation current is detected not in a mass-dependent manner, but it detects the oscillation of all of the ions. But what happens then is there's a mathematical transformation called a Fourier transform that is applied to this transient to convert that into a mass spectrum. And so this is a very different property, but it also a type of mass spectrometer, but it also has the property of a time of flight in that it can measure all of the ions in a population in one scan. The final one I want to introduce is called an ion trap, and it does lots of things. It's a very versatile type of mass analyzer in that it can store ions. You can actually introduce ions into the trap and store them until you do something else with them. But it can also be used to filter and ultimately to separate. And in these properties, it serves as a mass analyzer as well, measuring mass through the ability to eject ions from the trap to a detector in a mass-dependent manner. And so it will generally be used to measure all of the ions in the population as well. So the quadrupole tends to be a filter. Many of these others, we tend to be analyzing the whole population of ions. Now, when we're moving towards tandem mass spectrometry, we're actually using multiple mass spectrometry steps. So it's called hybrid mass spectrometry or tandem mass spectrometry. And this is actually how an instrument is organized and put together. So I find this a useful exercise because all of the properties that we're describing about tandem mass spectrometry are physical events. There's physical devices that do the separation, and these things happen sequentially in time. So I try to draw this in a way that reflects that physical reality. So oftentimes we have chromatography on the front of the instrument. This could be liquid or gas chromatography. We have an ionization source that converts molecules into ions. Once we have them ionized, they're brought into our mass analyzer region. And all of these last four boxes here are all under vacuum. So typically the first component of a hybrid mass spectrometer is either a quadrupole or an ion trap. And these are devices that can be used as ion filters, or they can be used to analyze the whole ion population. So a quadrupole can let all of the ions pass, or we can scan through all of the masses. An ion trap can trap all of the molecules and eject them sequentially. To do MSMS, we then have to have a way to fragment a molecule, and that is done with a collision cell. So the quadrupole or the ion trap will release ions into the collision cell where fragmentation occurs. Then we have a second mass analyzer. This can be a time of flight or an Orbitrap or an ion trap or a quadrupole. The second mass analyzer step is used to detect product ions. We have precursor ions and product ions. And depending on the application, one or more of these mass analyzers will be more useful than others. And then finally, we always have to detect those ions. So there's always going to be a detector at the end. Toward the end of the talk, we'll talk a little bit about ion mobility because it is used in tandem mass spectrometry, and I think it's a useful component to introduce to you all so that you can understand when the papers that you see refer to ion mobility based applications. And when you're thinking about where this sits, you can actually find examples of ion mobility devices either sitting at the front of the instrument between ionization and the first quadrupole region, between the quad and the collision cell, or between the collision cell and the second mass analyzer. So depending on the instrument you buy, that mobility device could be in any one of those locations. Now we'll take that information and we will apply it to understand tandem mass spectrometry. Tandem mass spectrometry is also called MSMS and is also called MS2. These are all effectively synonymous. You can use them interchangeably. Depending on what paper you're reading, you may see one or more of these, and they all mean the same thing. In practice, though, tandem mass spectrometry is usually serving one of two main functions. And these two main functions determine the two modes of operation and generally the instrument architecture that we're going to use to execute that MSMS experiment. The first application that we will use this for is for confirmation of molecular structure or elucidation of molecular structure. So this is a case where you might want to be generating an MSMS spectrum, which can be used to understand what a molecule is. So you have the accurate mass of a molecule and you want to understand what its structure might be. MSMS can help to inform on that. The way that this experiment tends to be done is that you isolate a precursor ion, generally with that first quadrupole. You fragment the ion in a collision cell, and then you monitor all of the product ions to generate an MSMS spectrum. The second main mode of operation and main function is to use tandem mass spectrometry in a highly sensitive and selective targeted analysis. And when we're doing it in this application, we're going to isolate the precursor ion, we're going to fragment the ion, and then we're going to monitor one or a few specific product ions. So you'll notice that the first two steps on these two main functions are the same. We're isolating a precursor, we're fragmenting it. The question that the only distinction between the two is really whether we monitor one ion or a few ions, or if we measure all of the product ions. And that really determines the utility of the instrument that we're using and the functions that are enabled through this acquisition method. And we're going to talk a little bit more about those here shortly. So in a structure confirmation or elucidation type experiment, we introduce a population of ions. If you have a pure compound, you can say, well, I've only got one compound, so I'm introducing one molecule. But there's always contaminants, and there's different ionization forms that can occur. But we can use our quadrupole to isolate, in this case, only the ion of interest. And so remember that quadrupole is a mass filter. And so if we introduce three types of ions, the yellow, the orange, and the green, the quadrupole only allows the green to pass. So only those exit the quadrupole in a manner that we can measure downstream. Those are then introduced into a collision cell. In a collision cell, we, in one way, shape, or form, add energy to the ion of interest, the precursor ion, such that it generates product ions. That is, the individual molecule breaks up. And in breaking up, we are looking at fragmentation product ions. All of those product ions come out of the collision cell. And we might use a time of flight or an Orbitrap after that to look at the population of product ions. Now, those product ions may contain still some precursor. If we fragment it at a relatively low energy, we can have some remaining precursor. So we might still see a little bit of that green mass present. If, on the other hand, we fragment really heavily, the green may go away completely. And so with the time of flight, we measure the masses of all of the product ions to generate this product ion spectrum that we see in the schematic on the lower right. That information then can be used in many different ways. And one of the traditional ways to use this is through something called spectral matching. So you can take that MSMS spectrum that product ion spectrum, and you can search it against a library, which you can see in the image here on the right, where that matching score, the match quality, can give you an annotation. If we take an unknown spectrum in the red at the top and we search it against the library, we get a really strong match to this riboflavin MSMS spectrum, which tells us that this compound is likely riboflavin. And so that is a traditional way to do it. MSMS is also used in proteomics applications for peptide sequencing. It can be used for polymer characterization to look at fragment ion masses and intensities to show you how a polymer is constructed by the way it falls apart effectively. This can also be used in manual interpretation. The manual interpretation is much more requires a much steeper learning curve, a lot more experience, but it can be done. And it's basically a process of trying to understand, given a putative structure, what are the fragment ions likely explaining in that substructure. In the past decade or so, there's also been a lot of tools that have been developed which can take these MSMS spectra in and do some sort of computational interpretation. So this isn't library searching in the traditional sense. Typically, what goes into these programs is an MSMS spectrum of an unknown compound and many structures. So rather than a spectral library, you're searching against a structure library. And these tools can take those structures and sort of infer what fragment ions are likely to derive from those structures and say which of the structures present is best explains the MSMS spectra that we observed. So that could be its own talk. We're not going to go into that in a whole lot of detail, but know that there are a lot of things that you can do with these MSMS spectra in qualitative studies. The second application that I mentioned is for using MSMS in highly sensitive and selective targeted mass spectrometry analysis. And in this case, we take that population of ions again. Again, we use our quadrupole to isolate a particular precursor of interest. We discard all of the precursor ions that we are not interested in, which can be either one or a few precursor ions, or it could be hundreds of precursor ions, depending on the complexity of the sample. We then fragment, just as we would have in a qualitative study, and we generate precursor and product ions from that precursor population. Now, rather than monitoring all of the product ions, as we did with a time of flight or an Orbitrap, we have another quadrupole behind the collision cell. And that second quadrupole actually is used as a filter again to allow to pass only a specific product ion that we are looking for. And so what we have with this system is a really, really selective mass filtering device, whereby you can filter on the precursor mass, you can fragment that precursor mass at a given collision energy to ensure that only the right things fragment. You can then look for specific product ions, and only the molecules that give rise to those particular traits, those are the only signals that ever reach the detector. And so this is what's called a tandem quadrupole type experiment, or a triple quad type experiment. The triple quad, for what it's worth, is the collision cell. And so it's generally not a quad anymore, which is why people have moved toward tandem quad rather than triple quad, but they're equivalent instruments effectively. And so you get a great deal of selectivity, and in doing so, a great deal of sensitivity. So you have much, much less noise in your background with these types of assays. And so this type of application is often used for measuring things like hormones in complex biological samples, things that are found at very low concentration in a complex background. Newborn screening uses tandem quadrupole type assays. Generally speaking, this is done through chromatography. And so in the process of using this selective detection, we do this same scan over and over and over again sequentially, such that we end up getting a chromatographic peak where we can monitor that product ion intensity over chromatographic time. And that peak area serves as our quantitative representation of that particular signal. So this targeted MS-MS assay can be applied in a few different ways. And I just wanted to introduce a few of those terms so that when you see this in the literature, you can have at least a working understanding of what this means. Selected reaction monitoring is the process that I just described, where you use your quadrupole to select one precursor, and you then have a collision event, a fragmentation event, and use the second quadrupole to monitor one product ion. That is single reaction monitoring. Multiple reaction monitoring will take one precursor and monitor two or more product ions. So it's the same concept, but rather than monitoring one fragment ion from a given precursor, it'll be multiple fragment ions. That could be 2 or it could be 10, depending on the type of analysis you're trying to do. There are two more sort of acquisition strategies for this targeted MS-MS that I will mention. You see them less frequently, but you see them enough that it's worth noting. Product ion scan is a situation where you use the first quadrupole to scan through a mass range. That might be mass to charge 200, 201, 202, all the way up to, say, 400. But the second quadrupole is always monitoring for the same product ion. And in doing so, this assay can be used to detect a particular common substructure across a bunch of different molecules. There's another application called neutral loss scanning, where you keep the mass difference between the precursor and the product the same, so that you might have for example, a lipid class, the phosphatidylethanolammines, they give a common neutral loss of 141 mass units. And if you have your first quadrupole, 141 mass units above your second quadrupole all the time, you can scan through all of the masses that may contain that particular phosphatidylethanolammine structure, depending on the different fatty acid units, and detect all of them in one type of experiment through this neutral loss scanning. The next step after our isolation event is dissociation or fragmentation. That is the process of taking that precursor molecule and breaking it into smaller fragment substructures, which generate fragment ions of different mass. They all have a common theme. You're in some way, shape, or form adding energy to an ion to induce bond breakage. There are many methods, and we'll talk a little bit about a few of these. So I just wanted to list a few of these. Collision-induced dissociation is probably the most commonly used of all of them. And this is also called beam type CID, or CID for short. I put a note here to say that the use of CID can be a little bit confusing on some instruments, in particular the thermo Orbitrap platform. CID is called HCD, higher energy collisional dissociation, because CID is also used to describe trap fragmentation. And it is very confusing, but note that most of the time CID refers to collision-induced dissociation. Electron impact is a type of fragmentation, which is also simultaneously ionization. We're taking a neutral molecule in that case, exposing it to a beam of electrons, which both ionizes and results in a fragmentation pattern. These electron-based fragmentation methods are becoming a little bit more popular for LCMS experiments as well, through electron transfer dissociation and electron-activated dissociation. You'll also see reference every once in a while to infrared multiple photon dissociation. I don't think there's even any commercial applications of that. And then the last one listed there is trap type CID, which is also sometimes called CAD, collisional activated dissociation. And we'll talk a little bit about a few of those. So in the ARC, we have access to about 4 of these. So collision-induced dissociation is on pretty much all of the instruments that we can do that are MS-MS on, that are LC coupled. Electron impact is on all of our GCMS systems. Our thermo Orbitrap system has electron transfer dissociation, ETD, and trap type CID, or CAD, collisional activated dissociation. We'll talk a little bit more about each of these 3 because they're relatively prominent in the literature these days. So we'll start with collision-induced dissociation. The basic premise here is that we bring ions into a collision cell. And typically we have done some sort of ion isolation so that we have the green ions coming in. What we do then is to introduce an inert gas. This is generally either nitrogen or argon, but every once in a while you see other gases used into the collision cell. We then apply a voltage across the collision cell from the front to the back so that we accelerate our precursor ions through that inert gas. What happens then, obviously, is you have collisions between the ion and the gas in the collision cell. When the energy added through those collisions is high enough, it results in bonds in that precursor molecule breaking and possibly rearrangement during that breaking process. This generates product ions through generating product substructures of the precursor ion. And here's just a very simple illustration of what that looks like. We're almost always actually have gas in the collision cell. So if we're doing an MS experiment, we have our green ion of interest going into the collision cell, colliding with, let's say, our red nitrogen molecule. It just continues on. It pushes the gas molecule out of the way and it continues on down the path. It doesn't fragment. If, on the other hand, we accelerate that, we get that green ion flying faster. When it collides with the gas, it'll break up into two or more subgroups. Now, in this example, we've got two fragments. If this was one fragmentation, one of those fragments is likely to be charged and the other one is neutral. This could also be multiple fragmentations, and this is two ionized forms of fragment ions. But whenever you have one charge on a molecule, then only one of the products will be charged because you can't get multiple charges from a single. And so the way that we use this then is we can apply a different amount of energy depending on the compound. And in this schematic here on the left, if we set our collision energy to 10 volts, which is relatively low, we send our green ion in. We still have mostly green ion coming out, but there's a little bit of fragmentation because we have added some energy. And we might see a little bit of our blue and a little bit of our yellow. As we increase the voltage potential across the front to the back of the collision cell, we get more acceleration, higher energy collisions, and we see less precursor ion and more product ions. And then if we increase further, we can make all of our precursor go away so that we only have the smaller product ions observed. So the bottom line here is that the more energy we apply to that collision cell via voltage, the higher the velocity of the ions traveling through there, the more energetic the collisions, and the more fragmentation we observe. You have the opportunity in these sorts of experiments to try to optimize collision energy depending on your goal, but oftentimes this optimization will be compound and application specific. But it is something that you can do. Tuning the collision energy is commonly done in a targeted focused assay. Electron transfer dissociation is another commonly used tool, probably, though much less frequent than collision-induced dissociation. It's probably the second most commonly used. And the mode of operation here is to introduce a radical anion into the trap with a target precursor ion. And this radical anion is generated with an electron beam. So you introduce a compound in which can be ionized by an electron beam to generate a radical anion that transfers the electron to your target analyte. And so your target analyte is already charged, but in accepting that negatively charged electron, its charge can be distributed through that the mobile electron phenomenon and generate fragmentation in a way that can be very complementary to CID. So the fragmentation spectra for CID is different than that for ETD, and this is one of the reasons why it is used. One of the early applications of this was actually in the analysis of phosphopeptides. So if you look at the spectrum on the top, this is actually not CID, this is collisionally activated dissociation in an ion trap. But CID spectra would be similar. When you fragment these phosphopeptides, what you tend to see is the loss of this phosphate group and not much else. And so you don't get a lot of information on the peptide backbone sequence, just that there is a phosphate. When you use an ETD, electron transfer dissociation type method, you get much more structurally relevant fragmentation spectra, where you can see the backbone fragment ions based on and be able to infer the amino acid sequence and tell you that it's phosphorylated in one spectrum. And so there are certain types of molecules in particular that ETD tends to be used much more for. Electron activated dissociation, EAD, is a relatively newly commercialized approach. It's kind of similar to electron transfer dissociation, except that we don't have a dedicated reagent ion that we ionize first. We actually irradiate the ions that we have formed through our ionization source in the front of the instrument with an electron beam to directly add electrons to those structures. Now, this is often a combination of what's called ETD, electron transfer dissociation, and ECD-like mechanisms, where ECD is electron capture dissociation. But basically, the basic premise here is that you are adding electrons to an ionized molecule already. That radical fragmentation pattern can be more structurally informative at times than the collision-induced dissociation fragmentation pattern. The downside to this is that it can be appreciably slower than CID, and we'll get to why that's important later. And it's relatively hard to get on commercial instruments. As far as I know, this is only available on SciEx QTOF type platforms. And I bring this up here. So electron impact is the method that we use for gas chromatography. This is effectively the same thing as electron activated dissociation, where we're irradiating rather than a ion in EAD, we're irradiating a neutral molecule with electron impact, which ionizes and causes fragmentation at the same time. So, but there is actually a lot of overlap between electron impact and EAD in principle. Okay, so I wanted to go through the hardware, and now the question is, how do we use it? I'm going to break this into two sort of sub-discussion areas. One is in talking about precursor selection. The first is the approach, and how do we use the instrument to select precursors? And we're then going to talk about the actual hardware used to isolate precursors. So on the first side, the approach, what I'm referring to here is, are we going to select precursors in a predefined manner, which is what I will call targeted? And if we are doing a targeted sort of tandem quad assay, we want to measure hormones, we are going to tell the instrument what the precursor is, what the retention time is of our analyte if we're using chromatography, what collision energy we want to fragment at, and what the product ions are. So all of these things are predefined. Before we make an injection, the instrument has a to-do list that it will follow. That contrasts with an untargeted approach, where we were not telling the instrument necessarily exactly what we want done. We're not saying, go look for this molecule. Instead, we have two different approaches, which we'll get into the details on later, called data-dependent acquisition, where precursors are selected based on the data that is being acquired. And so you give it sort of a list of rules before you make the injection. But the decision tree is dependent on the data as it's being acquired. And a second approach called data-independent acquisition, which is actually rescheduled, but not targeted to any particular molecule. And I'll explain what that is as well. The second subcategory that we'll talk about is the precursor isolation method. And this comes down to the hardware. You can use a quadrupole or an ion trap or ion mobility to select precursors. And we'll talk about a little bit about what those look like. So in targeted precursor selection, this is the type of application that we would use in a multiple reaction monitoring experiment, single reaction monitoring experiment on a tandem quadrupole instrument. The way this generally works is we tell the instrument in advance that we're looking for that green precursor ion, and we're now monitoring two product ions, a blue one and a brown one. Of course, they're not colored in real life, they're different masses. But we take those two product ions and we look at their intensity as it varies over chromatographic time to give us these chromatograms, extracted ion chromatograms, or product ion chromatograms, whereby we can quantify based on the peak area of our signals. And we can also use the multiple signals to confirm that we're targeting the right compound. For a given compound, the ratio between the blue and the brown curves will be very constant. If we see deviations in that ratio, it can tell us that we may be picking up signal from another compound. But we can also use targeted precursor selection in a qualitative experiment. So if we have one or a few compounds that we want to generate high quality MSMS for to help confirm structure, we can tell a QTOF type instrument that we want to select this precursor at this retention time and collect as high quality an MSMS spectrum as we can. We can give the instrument as much time as we want because it's the one thing that we're trying to do with it. And so that can be a very targeted experiment as well. And that's a case where we would tell the instrument in advance specifically what we want it to do, and we're trying to look for one or a few things. In an untargeted type application, the most common approach for collecting MSMS data is called data-dependent acquisition, DDA. You will see this abbreviated in the literature and on websites. And I'll walk through the principle of this. So the first thing that you're doing in a DDA experiment is you're actually collecting MS level data. So you're acquiring a scan, which I call here MS1, which is not a scan of product ions, but of all the precursor ions that are present. And you can see what this looks like up in the upper right-hand corner here. So this precursor ion spectrum, or MS1 spectrum, in this case, we have 6 different ions that are detected. Each of them is colored differently in this case. So what DDA does is then selects one or more of those precursor ions to fragment. In this case, it will select 2. We've told it to select the top 2, so it selects the brown one, which is the most intense, and this blue one, which is the second most intense. That decision has occurred after this spectrum has been collected, and then immediately the instrument moves to acquire an MSMS spectrum on the data-dependent precursor chosen for this brown molecule. It collects an MSMS spectrum where we fragment it, and then immediately after that collects an MSMS spectrum on the blue one. And so the data-dependent MSMS component is because this spectrum, the mass to charge and intensity values in that spectrum determines what happens next. And so in this way, you can actually make an injection of a sample, and without knowing what you're looking for, try to collect as much high-quality MSMS data as you can on all of the signals that are present. So it goes through this cycle. It has selected the first 2, performed MSMS on each of those 2, and then it collects another MS scan. After it has collected the first 2 most intense, we can set a rule to say don't sample those again for at least another 5 seconds or 10 seconds. And so then it selects the next most intense, the purple, and the next most intense, the black, and performs MSMS on those. And through this process, you can get relatively deep coverage of all of the major signals that are present in a way that you don't have to define what signals you're looking for. This tends to be performed on time-of-flight and Orbitrap-based systems, QTOF type applications. But with an Orbitrap in particular, they're often coupled with ion trap. And so these applications are very widely used in proteomics and metabolomics experiments because those MSMS spectra are really valuable for assigning structure to ions. Once you have those spectra, they are essentially the same as the targeted spectra that we described above. And so in the prior slide, we said we could have told the instrument, go look for that brown ion. We know its mass to charge ratio, we know its time. If we knew that 20 volts was optimum collision energy, then we go have it collect that. And we could use DDA in the same approach and say, look at everything, but fragment anything you see at 20 volts. The product ion spectra would be identical, given signal intensity is sufficient. And so you can use these DDA spectra in the same way that you would use a targeted MSMS spectra. The benefit of a targeted approach is often that you can get higher quality data. And when I say higher quality, higher signal to noise, because you can dedicate more time to it, you can be more focused in how you acquire the data. It just takes much longer to do it that way and can be more expensive in the end. There's another concept we have to introduce here before I switch to data independent. So when we're doing data dependent acquisition, I mentioned that the algorithm selects the brown ion, and that we might have measured with a high mass accuracy time of flight mass spectrometer. So in that type of mass spectrometer or an Orbitrap, our mass error is under 10 parts per million mass error. But you have to remember when you're doing these experiments that it's actually a quadrupole or an ion trap that's doing the precursor selection. And they are not accurate mass instruments. And so what we have is we have a set mass that the quadrupole is isolating, but it's also isolating a small range of masses around it. And so I refer to it as the window here. So this mass to charge space on either side of your target ion, this could be anywhere from about 0.4 to 4 mass units, depending on the instrument and the settings that you're operating under. So in the example that I have here in the spectrum, our isolation window, which is this gray box, isolates the blue ion and excludes the purple. The MSMS spectrum that we generate then is going to have fragment ions that are only derived from the blue precursor. However, if our two masses fall within the isolation window, so even if we trigger on the blue mass, the isolation window is wide enough that we get the purple ion transmitted through the quadrupole as well. That means we're also going to fragment the purple ion, and the product ion spectrum is going to contain signals from both the blue and the purple precursor. This is commonly referred to as a chimeric spectrum. We have multiple fragment ions from one MSMS precursor selection event. It represents 2 molecule fragmentation patterns. And of course, when you get this out, these aren't color-coded. So it can be difficult to know which fragment ions are derived from which precursor. There are ways to do that, but again, that's a very much more complex talk than we're going to cover today. And so MSMS speed becomes important, and I'll tell you why. In an untargeted, data-dependent experiment where you're performing MSMS on as many signals as you can, the more complex the sample is, the more precursors need to be sampled. Every one of those MS events take time, and these are chromatographic peaks, so they don't last forever. You might only have a few seconds to sample a given precursor. If there is not enough time to sample all the precursors, then you don't get MSMS data on them, and you can't identify them with any confidence. And so this is a case where MSMS speed can prevent you from performing a very detailed characterization of what's in your sample. You might get the tip of the iceberg depending on how complex the sample is. In a targeted experiment, every MSMS transition takes time. You need at least 10 points across the chromatographic peak. The more transitions you have, the less time you have available per transition to keep enough points across the peak. And you'd start losing sensitivity because the more time you spend scanning a particular ion, the more sensitivity you have. And so speed is really important. And as technology has developed, and as applications have become more complex, and as biologists want to see deeper and deeper, and environmental scientists want to find lower and lower level trace level contaminants in environmental systems, it has pushed the limits of what data-dependent acquisition can do. Data-dependent frequently becomes too slow to be utilized for comprehensive coverage. That has resulted over the past decade or two in the development and adoption of an MSMS approach called data-independent MSMS. And basically, the concept here is that we are not going to make MSMS dependent on the MS1 spectra anymore. Instead, we are going to set up an MSMS method that samples all of the masses all of the time. And the way that it does this is it scans through a particular mass range. In this case, in this table, you see a start mass of 50 and an end mass of 1,000. Now, within this first bin, this box, we actually set the range rather than to say 4 mass units. It could be as much as 50 or 100 or 1,000 mass units. And so we accept these wide windows, acknowledging that all of the fragmentation spectra are going to be chimeric. But in setting it up this way, we scan all of the precursors between 50 and 1,000 every scan cycle. So we can guarantee that coverage is good, that we have sampled everything that is detected, even in complex samples. The penalty that we pay is that the spectra are more complex. The MSMS data are now very complex. We have to assume that there are product ions for multiple precursors in every MSMS spectra, and it usually requires dedicated informatics to do spectral cleanup. There's also different versions of this data-independent MSMS. The one that I show in this graphic on the right is basically what's called SWATH. This is a product that was marketed and developed by SciEx. And it's basically a sequential process where you step through mass ranges, one by one, all the way through from the lowest mass of interest to the highest mass of interest. And then you do it again, and then you do it again. And that whole cycle might take a second, or a half a second, or 2 seconds, depending on your instrument. And then it does it again, so that you get complete coverage throughout the entire chromatogram. Now, there's a trade-off here between sensitivity and selectivity. So as I have this drawn, I think I have 20 mass bins here. You could do 200 mass bins, so that every one of these ranges, rather than being, say, 50 to 100, is more like 50 to 60. So you have less chimeric risk with that, but you have much less time to spend because you have more steps that you need to do. So there ends up being a really explicit trade-off between sensitivity and selectivity with this DIA type approach. So DIA, as a quick overview, sacrifices MSMS specificity to get improved coverage and speed. And we use these in metabolomics approaches. We use these in proteomics approaches. It does require special data processing. The spectra that you develop, even though they may contain all the same signals as a traditional DDA type spectrum or a targeted MSMS spectrum, can't be handled in exactly the same way. So we'll use special software and data analysis approaches to deal with that. This has become really popular in proteomics because it's enabled much higher data acquisition speed while retaining protein coverage, proteome coverage. And so you'll see applications in the literature looking at 5 or 10 minute chromatography rather than 1 or 2 hour chromatography as you often have with DDA. There are trade-offs with it, but it's enabled higher throughput, which is particularly appealing in a lot of clinical assays. Now I want to cover one final topic here, which is a little bit of a diversion, but it's actually become prominent enough that I think it's something we got to cover when we're talking about MSMS. Ion mobility is a separation modality that operates, again, in the gas phase. So this is a device that is under vacuum after ionization. After things have been introduced into the mass spectrometer, we can introduce an ion mobility cell. Again, this is something you buy with the instrument generally. This mobility device separates those ions not by their mass, but by their shape. And shape does correlate to size. A small ion of low mass will have low volume as well, and that impacts the shape. But an ion of high mass will have a high volume, and that impacts shape. But you can have 2 ions that have the same mass, where their ion mobility shape is a little bit different. So you can see then how that might be valuable. If I say they have the same mass, you as chemists might be thinking isomers. And so you might have two molecules that have the same formula. Mass spectrometry can't isolate those two things from each other. Ion mobility sometimes can. They aren't as high resolving as mass spectrometry, but they can perform mobility-based isomer separation. There's a lot of different types of ion mobility spectrometry, IMS. We're not going to cover all of them, but they all can be used. And the reason I wanted to introduce this is because ion mobility can be used in precursor selection for MSMS experiments. And it does so by using the property that ions are focused by their mobility. And there's effectively two popular IMS configurations that I wanted to introduce here because you will see them as you're in the literature. The first one you can find on Bruker mass spectrometers. It's called PASSF, Parallel Accumulation Serial Fragmentation. And this is found on what's called their TIMS TOF. TIMS is Trapped Ion Mobility Spectrometry, time-of-flight platform, so TIMS TOF. And basically, it's a faster, more sensitive DDA. So this is the instrument architecture. So if we look at the configuration, what we have here is we have an ion mobility cell placed in front of the quadrupole. So normally this wouldn't be here. We would have our ionization source right here instead. In the TIMS TOF, the ionization source sits up here. Mobility is the first separator. And in trapped ion mobility, it traps the ions at the front and then releases them based on the mobility. And so what happens then is you have ions getting focused. What would normally be a continuous beam of ions coming out, we now have focused by their mobility. And so the early eluting ones, when we're doing data-dependent acquisition, we can now collect a MSMS spectrum and say sample 10% of the ions in a fraction of the time that it would take normally in DDA because we have focused them in time. They come out of the mobility cell focused by time, so the quadrupole then can spend less time on any given ion and still have the same signal intensity. We can get more sensitivity with this approach by focusing the ions and syncing the quadrupole to the mobility. In doing so, we can spend less time on every MSMS event. And in practice, you can collect MSMS spectra anywhere from 5 to 10 times faster and retain data quality for every one of those MSMS spectra. And so it is a way to make DDA faster and more sensitive. It's really only available on this TIMS TOF platform, but it is widely used in the proteomics field. Here is another application using mobility. This one is called HD-MSE. MSE is a process that was marketed by Waters. You fragment everything all the time by literally not isolating any precursors whatsoever. And so the quadrupole is passing all of the precursors. When they then introduced ion mobility into their platform, they introduced the ion mobility cell after the quadrupole such that you can separate by mobility and then fragment all of the ions coming out of the mobility cell as they elute in the collision cell and collect fragmentation data on all of them all of the time. And so what we do then is we actually use the mobility cell as the precursor selection device. So the focusing by mobility is performed rather than a quadrupole-based isolation. And this is a form of data-independent acquisition using mobility. So those are more complex. We can certainly talk more about them if you have questions. But I wanted to introduce them at least a little bit briefly because they are more and more frequently used in the literature. And now to summarize, we've covered a lot about MSMS, starting with the notion that tandem MS is enabled by hybrid instrumentation. You can't do tandem MS without the ability to do a precursor selection and a product selection separated by a fragmentation. And so most of the time that is through coupling two mass spectrometers. I didn't talk too much about ion traps, but ion traps can actually do this sequentially in one device. MSMS can be used in very sensitive targeted assays. So this would be the application of things like single reaction or multiple reaction monitoring, but it can also be used in qualitative studies to assign structure and improve confidence in identification of unknown structures. There are a lot of different ways MSMS can be used. And we talked about the various precursor selection mechanisms that include both quadrupoles and ion traps, but also ion mobility. Precursor selection approach can be very targeted or very untargeted. That is, we can be very focused on one ion if we have a very targeted question, or we can try to be as broad as possible by using a data-dependent MSMS approach where we're still selecting precursors, or data-independent approaches where we're selecting a wide range of precursors without any notion of what the MS level data looks like. And finally, we talked a little about the various dissociation methods. Collision-induced dissociation being the most prominent, but some of the electron-based dissociation tools are becoming much more widely used and are worth keeping an eye on moving forward. And with that, I wanted to open it up to questions. I see a hand up from Jacob.
Hey, Corey, excellent talk. I have a question about the non-target aspects of this. So you're talking about how as you alter your collision energy, that will cause your fragmentation spectra to look different. You know, more higher collision energy, more fragments, less precursor ion. When you compare those fragmentation spectra to a library database, do you have to take that collision energy into consideration since one precursor ion could have different looking fragmentation spectra depending just on what that collision energy was?
It's a very good question. So the MSMS libraries, depending on which one you're searching, the example that I showed in the screenshot was from the NIST, the National Institutes of Standards and Technology MSMS library. And when they build their library, they actually generate the library with many collision energies. So they might collect collision energies at 5, 8, 11, 14, 17, 20, 23. So very fine increments so that it's very likely that any spectrum that you generate is actually going to be represented very well in the library. But it can be an issue if the library isn't developed in that manner. If your collision energy is at 30 and the only library one is at 10, then the quantitative differences are going to be relatively large in the product ions that are detected. And sometimes you might not get the same ions at all. Oftentimes there's still enough overlap, particularly if you're using accurate mass MSMS, that you can say, well, it looks like it's probably this, but the spectrum match isn't great because the product ions are qualitatively the same, even if the quantitative data isn't exactly the same.
Thank you.
I see a question from Joe. Hand up.
Hey, Corey. Yeah, thank you for the great presentation.
Mm-hmm.
And I have one question and for the differences and between QTOF and Orbitrap on the untargeted precursor screening. So do you think you can give us just like some kind of general information about the pros and cons for these two types of instruments and on the untargeted precursor selection?
They're actually not that different. So there's Orbitrap is actually a sort of a family of instruments, but if we think about a quadrupole coupled Orbitrap and a quadrupole coupled time of flight, they're actually pretty similar. Their performance specs aren't that dissimilar at all. An Orbitrap, you can in theory get higher mass resolution and higher mass accuracy than on some TOF platforms, but it comes at the expense of increased time per scan. And so there's a trade-off between mass accuracy on that platform and scan time, which doesn't happen on a time of flight. Orbi spectra can often be cleaner because it goes through a mass or a mathematical transformation before you see the spectrum. But ultimately, the properties are pretty similar, especially since Thermo has implemented their HCD or CID-based fragmentation. You can find a lot of strong opinions out there. Most of the time, I don't think that they're warranted. There are definitely times where an Orbitrap is going to have properties that a TOF doesn't, and there are times when a TOF is going to have properties that an Orbitrap doesn't. So under certain applications, one might be better than another, but generally speaking, they both have enough really strong qualities that I would be hard pressed to pick one over the other for general application.
I see. Yeah, thank you.
Mm-hmm. Other questions? Okay, one final reminder then to everybody that that hopped on a little bit after I started. We do have another talk February 2nd from Dr. Claudia Boot using mass spec and NMR for metabolomics. And March 12th, Dr. Ethan Crace on twinned crystal analysis and X-ray diffraction data. So please look those up and sign up if you have interest. If you do have any further questions on MSMS, don't hesitate to reach out. My name's all over our website. Thanks everybody for coming.
Yeah,
Thanks a lot, Corey. That was really great talk.